Now, after making all the arrangements, we are going to perform streak plating. How to do this method? सबसे पहले जो काम आपने करना है, जितनी भी पेट्री प्लेट्स आप यूज़ करना चाहते हैं, 2, 3, 4, उनके ऊपर अपना एक लेबल लगाना है. That label contains your name, date, class, and the source of culture which you are going to use for streak plating. This is very important to identify your cultures in the later stages of your coursework. Streak plating के दो डिफरेंट तरीके हैं. एक को हम क्वाड्रेंट स्ट्रीक कहते हैं और दूसरे को हम कॉन्टिन्यूअस स्ट्रीक कहते हैं. I will suggest you to prepare 2 plates for quadrant streak method and 2 plates for continuous streak method. आइए अब हम देखते हैं दोनों तरीकों में क्या फ़र्क है. Quadrant streaking and continuous streaking methods, they are not much different. जैसे कि हम इस स्लाइड में देख रहे हैं, 2 पेट्री प्लेट्स और उनमें स्ट्रीक्स हमारे सामने हैं. क्वाड्रेंट स्ट्रीक मेथड में हम पेट्री प्लेट को वर्चुअली 4 हिस्सों में तकसीम करते हैं, लाइक दिस, और हम हर एक हिस्से में एक अलग से स्ट्रीक बनाते हैं. क्वाड मीन्स 4, so that is the reason this method is called as quadrant streaking. Once you make a mind of virtual division, then using the inoculum loop, you can move up and down freehand, right or left freehand at your convenience. Keep in mind, while doing streaking, maintain the sterilized conditions. Don't remove the petri plate cover totally. Don't touch the petri plate cover to the surrounding bench or any other piece of equipment. Continuous streaking is a complete plate. We simply use it for streaking of the inoculum like this, right or left at your convenience. Then holding the plate, rotating it to 180 degrees centigrade, making another streak. Make it sure don't take too much sample in the inoculum, in the loop, because if you take too much sample, then you have to do many streak preparation, then you have to do many streakings so as to get a single cell isolated on the agar plate. The next step towards streaking is taking the inoculating loop and making it sterilized on the flame, as you can see here. Wait until the loop becomes red hot. Let the loop cool down. Don't wave the loop in air. Don't touch the loop with anything to make it quickly cool down. If you do this, then you are basically going to get again the unsterilized conditions. Once your inoculating loop is sterilized, take the sample from the inoculum. Once your loop is sterilized, take the inoculum from the culture. Carefully touch the culture containing loop with the agar plate and make your streaks. Like अगर आप इस पर्टिकुलर पेट्री प्लेट में देखिए, डिफरेंट स्टाइल से स्ट्रीक बनाई जा रही है, और अगर इसको ऑब्जर्व करें, देयर आर मल्टीपल स्ट्रीक्स. स्ट्रीकिंग करते वक्त एक चीज़ का ख्याल रखिएगा, डोंट एक्सर्ट एक्स्ट्रा प्रेशर बिकॉज एगर इज अ जेली लाइक material. It is not as hard as cement. So if you are going to put extra pressure, you are basically going to destroy the agar plate. If you have to do another streak again, follow the same procedure. Go to the flame, sterilize the inoculum, sterilize the loop, cool it down, take the inoculum from the culture and streak again. So the continuous streak method, using the same procedure without any quadrant, you simply make the streaking. For control, Don't take any inoculum and repeat this process with simply sterilized inoculum loop. And after that, incubate your E. coli containing plates at 37 degrees centigrade and the yeast containing plates at 30 degrees centigrade for 24 to 48 hours. And after the given time, observe your plates. You should come across with the isolated colonies towards the end of your streak.